So this is the, excuse me, this is the presentation on more Punnett square notes. We'll be discussing things like dihybrid Punnett squares. We're looking at two different traits. We'll be looking at how to construct the dihybrid cross, some alternative genetics. We often say these are the exceptions to Mendel's rules, where we talk about incomplete dominance and codominance and sex-linked traits and multiple alleles and blood types and polygenic inheritance. So as we go through the presentation, you should have the notes sheet out in front of you. If you feel comfortable taking your own notes, feel free. I highly recommend you using the class notes. If I go too fast, just make sure you stop and you stop the presentation so you can write down what it is you need to write down. And of course, it wouldn't help to come back and listen to this again if as a means of studying. But if we start with dihybrid crosses, and previously in class we've been working on the monohybrid crosses, the single trait crosses, where you have a 4-box Punnett square. And again, the Punnett square is just looking at the possible outcomes of offspring. And so we're going to do the same thing here, only we're going to be looking at two different traits. For example, we'll have a big R and little r and a big T and little t. So these are combined traits. So looking at the laws of segregation and law of independent assortment, the law of segregation states that only one allele for each trait will be passed on to a gamete. Now we learned this when we talked about Mendel. Only one allele for each trait will be passed on to a gamete. So you get half of your genetic information from each parent. The law of independent assortment states that the alleles separate independently. Now you have two alleles for each trait. They separate independently, and only one of them gets to be passed on to each gamete. For instance, you have a diploid parent, but when the gametes are made, there's going to be always, in each gamete, there will be one R and one T. For instance, you could have the possible combination of a big R and a big T in a gamete. You could have a possible combination of a big R, little t in a gamete, little r, big T, or a little r, little t. These are all the possible haploid gametes. So if we look at these options, there are 4 possible options for each parent. So our Punnett square is going to be a 16-box Punnett square. 4 possibilities from each parent. So if we look at this chart here, these are the possible genotypes. And again, here's our example in pea plants. Round seed is dominant over wrinkled, and tall plants are dominant to short. So if we look at 2 pea plants, both heterozygous for each trait, what are the possibilities of the genotype and the phenotypic ratios of their offspring? So moving back a bit in your notes, you have parent genotype. Big R, big T, little t. What is the possible gamete? What's one possible gamete? Go ahead and fill in the chart for each of the possible gametes. What could be a possible gamete here? What could be a possible gamete from this combination? What could be a possible gamete from that combination? So go ahead and fill in your notes, and then we'll again go back and start working on how to construct our dihybrid cross. So in class, I'm going to check your possible gametes to make sure that everyone is on the same page. So if we move to our next session, constructing a dihybrid cross, and we look at our example again, round is dominant to wrinkled and tall is dominant to short. And so we have a situation where both parents are heterozygous for each cross. So we want to take a look at, if it's heterozygous, then one possible combination is going to be big R, big T right here. Another possible combination would be big R, little r, big T, and big little r, little t. And so each parent has those possible combinations. So it's a 16-box Punnett square. One parent goes on the side, one parent goes on the top. And if we look at this first box here, each parent donates a big R. R's go together. Each parent donates a T, big T. Big T's go together. Now remember, in these boxes, the R's go together and the T's go together. You would not write this as big R, big T, big R, big T as the individual. R's stick together, Those are the alleles for one trait, and the T's stick together. Those are the alleles for another trait. So in the next box, what would the next box be? Right here. Big R, big T, little t would be the next box. So you fill in each box, and then we get the next things to figure out the ratios. So if we did every single box, this is what you would get. And this is the hardest kind of dihybrid cross, two heterozygous parents. And so we first take a look at the genotypic ratio. Now if you take a look, genotypic ratio, and I'm going to look at the first box. You always start to look at the first box. How many big R, big T's do you have? Just one. Big R, big T, little t's? You have two. One, two. Big R, big T? One, two. Big R, big T, little t. One, two, three, four. Now, what I would do, and I don't have the option here, my computer isn't allowing me to cross these boxes off, I would cross off your boxes. So the next box is big R, or little t. One of those. Let me go here. Big R, little t. One, two. Little R, big T. One, two of these, and then one of those. So our genotypic ratio, moving back here, is a 1 to 2 to 2 to 1 to 4 to 1 to 2 to 2 to 1. It's a long ratio. So again, the genotypic ratio is just stating what are all the different letter combinations. And if you want to check yourself, you should add these numbers up and they should equal 16. So 1 plus 2 is 3, plus 2 is 5, plus 1 is 6, plus 4 is 10, plus 1 is 11, 13, 15, 16. My numbers add up to 16. So the phenotypic ratio, what do these letter combinations give you? So a big R, big T is going to give you a round and tall. This is going to give you a round and tall. So we'll give you a round and tall, round and short, round and tall, wrinkled and tall, round and short, wrinkled and tall, and wrinkled and short. So if you add up these numbers, these are all the different individuals with those particular phenotypes, you would have 9 of them would be 9 smooth and tall, 3 would be smooth and short, 3 would be wrinkled and tall, and 1 would be wrinkled and short. So before we move on, I want to do one more example of a dihybrid cross. Okay, just making sure that I'm recording here. Let's go ahead and here we go. Here's another example. Okay, dealing with horses. In horses, black hair is dominant over brown hair, and trotting is dominant to pacing. Cross 2 parents, heterozygous for each trait. So then this is just another example. You don't have to write this down if you don't want. If you would like to write this down on the back, you certainly can. But if each parent is heterozygous, we have male, we have the female. For the male, a possible combination for a sperm could be each sperm is going to get a B in either a big B or a little b. So one combination could be, and I'll take a look, one possible combination could be big B, big T. Another possible combination could be big B, little t. Another possible combination could be little b, big T. And the other possible combination could be little b, little t. And of course the other parent, I'm going to click through here, that's a combination of an egg, that could be another combination of an egg, another combination of an egg, and another combination of an egg. So when you put these together in a Punnett square, okay, it's thinking here. There we go. We have a 16-box Punnett square. Sperm are on the side, eggs are on the top, and each parent would donate a B, each parent would donate a T. Each parent would donate a B, each parent would donate a T. So then you would fill this in. There we go. There's our Punnett square. So if we look at the genotypic ratio of the genotypes, again, you might want to start making a list here. You have how many of those letter combinations do you have in the 16-box Punnett square? You've got one. Cross it off. I said that in the last one. I would recommend crossing it off. In this, you would have 1, 2. And notice that you're going to see a pattern that exists here. 2 of those. Big B, little b, big T, big T. 2 of those. Big B, little t, both heterozygous. 4 of those. 1 of those. Big B, little t, little t. 2 of those. 1 of those. 2 of those, and last, only one recessive individual, a brown trotter. So, black trotter, black trotter, black trotter, black trotter, black pacer, black pacer, black trotter, or sorry, brown trotter, and brown pacer. So if we look at our genotypic ratio, 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1 ratio. And if we want to look at our phenotypic ratio, how many black trotters do we have? And a lot of people would say we have 4 black trotters, but that's not the case because we have to add up all these numbers here. So that equals 9 black trotters, 3 black pacers, 3 brown trotters, and only 1 brown pacer. Genotypic ratio, phenotypic ratio. There we go. We won't do that. We'll save that for class. So we'll go back here. If you have any questions with the dihybrid crosses, again, you can certainly see Mr. Matthews. But going on to alternative genetics, and this is mostly, this is mostly we call exceptions to Mendel's rule. Incomplete dominance is the first example. So these are individuals that are heterozygous and have blended phenotypes, mixtures, a mixture. So for example, we have snapdragons and snapdragons. And it looks like just substitute, I'm sorry in the notes, just substitute N's for R's here. Big R would be red, little r would be white, and big R, little r would be pink. So if you cross 2 heterozygous snapdragons, the male is heterozygous, the female is heterozygous. So you would have a male that is pink, a female that is pink, and there's a possibility they could have a red offspring or a white offspring. About a 25%, 1 out of 4 chance they could have a red offspring, and a 1 out of 4 chance they'd have a white offspring. And then, of course, a 50% chance they would have a pink offspring. So this is incomplete dominance. All right, there is no necessarily dominant allele here. If we look at something called codominance, right? In horses, heterozygous expresses both traits. In horses, red and white equals a roan color. And another example of codominance is in chickens, where we see both kinds of fur. So again, a big N is going to be black, a heterozygous would be checkered, and little n would be white. So if we do the Punnett squared, the right of your notes, if you take 2 individuals that are checkered, there's a chance you get a 25% chance you get a black individual, a 25% chance you could get a white individual, and a 50% chance you'd have a checkered individual. So again, in your notes, it would be good to list the genotypes and the phenotypes. Go ahead and do that. And if you haven't done that for the snapdragons, also do that for the snapdragons in the example in letter B of your notes. So this is incomplete dominance. This is one exception to Mendel's rules. If we look at sex-linked traits, traits that are sex-linked are carried on the X chromosome. Right? The Y chromosome doesn't have the allele, so females would have how many? How many X's do females? So females have 2 alleles for each gene, while males only have 1 copy. So males either have it or they don't. Females either have it if they could be a carrier, or they don't have it. So you generally see males with more of a chance of getting or having these sex-linked traits than females. So there are some examples. Color blindness, hemophilia is a blood disorder where your blood does not clot and you can bleed to death internally. Just a simple bruise could make you bleed to death. That's not very good. So if we look at the X and Y must be included in the Punnett square, but the X chromosome does not have, I'm sorry, the Y chromosome doesn't have an allele. So that's something to keep in mind. You must put the X on but Y does not have the allele. So if we look at, for instance, the phenotypic ratio indicates if the individual is male or female. So if we look at hemophilia, and hemophilia is person lacking blood proteins for clotting, and so you could have someone that could get a bruise and not be able to stop it from bleeding, I'm sorry. And so if we look at what it's caused by a sex-linked recessive allele. So if we look at the different genotypes, a completely homozygous dominant female would be normal because hemophilia is a recessive allele. So an individual that's female, where both of their X chromosomes are dominant, would be normal. If they were heterozygous for it, they would still be normal, but they would have a chance of passing that trait on to their offspring. So with the female, the only way to have hemophilia is for both of their X's to have the recessive trait. So there's a 1 in 3 chance that a female could have it. With the males, because they only have 1 X chromosome, they're either normal or they don't have it. Chances are that most people in this classroom are not hemophilia. So if we look at a genetic problem, a man that is normal has a child with a woman who is a carrier. What is the chance that the child would have hemophilia? So of course, the female is a carrier. The male is not. So you'd set up the Punnett square. X, 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 Y, and in the box here, again, each parent is going to donate half. So the question is, again, what is the chance that the child would have hemophilia? And the answer is 25%. 25%, 1 out of 4. Now, if you look at the males, right, there's about, if they have a boy, it's about a 50% chance, of course, that they would have a child with hemophilia. So these are examples of sex-linked genes. Another example of exceptions to Mendel are multiple alleles, and this is what we deal with blood types. Individuals only inherit 2 alleles. We deal with blood types here. So in this case, we have 2 alleles that are actually dominant over the other. 2 alleles that are dominant over the other. Bring back here. Make sure you get this information down. Example blood type. So if you are type A blood, you can either be type A, type B, type AB, or type O. You're possible, and we're going to use capital I as dominant and little I as recessive. And the capital I can either be the A allele or the B allele. If you're type A, you can either be homozygous, IA, or heterozygous, IA little I. If you're type B, B is also dominant, 2O. You can be homozygous or heterozygous. If you're AB, then you have one of each. If you're O, you are recessive. Again, this is mainly just a type of protein on your red blood cell that determines what kind of blood typing you are. And so if you're type A blood and someone gives you B blood, your body won't recognize these proteins and you'll reject that blood. If you're type B blood and you're given type A, your body won't recognize it and will reject it. If you're type AB, you can receive A blood, B blood, or even O blood. That's what they look like under the microscope. We'll talk more about what anti-A and anti-B mean. We actually have a lab that goes along with this. But if we look at if a type O male, I'm going to pause it here. Give me a second. All right, we're almost finished here, and just want to go back to what we were talking about. Okay, we have a individual that a type O male and a homozygous type A female produce offspring. What are the possibilities of blood types of their children? And this comes in very, very useful. I actually did this with my own kids because I know that I'm type A and my wife is, I'm type AB and my wife is type O. So I was able to figure out what exactly my children would be when we were sitting in the hospital. And this comes in handy if you ever need a blood transfusion. Typically, though, if you're in an emergency situation, doctors will, because most people don't know what blood type they are, and if you're in an emergency, you don't have time to really do a blood test, you'll just be given type O because most people can't accept whatever blood type they are. You can't accept type O blood. So if you're type O, and that's about 50% of the people on this planet, you're considered the universal donor. I'm type AB blood, and anyone that's type A, it's the rarest kind of blood. We are the universal receivers because we can receive blood from anyone. Type O people can only receive blood from type O. Their bodies will reject A, it'll reject AB, it'll reject B. But that shouldn't be too much of a problem because 50% of the people on this planet essentially are type O, so it's not too big of a deal. But back to our problem here. We have a homozygous female that is IA, and we have a male that is type O. What are the chances, again, what are the chances, or what are the possibilities of their children? Male can donate no matter what. He's always going to donate the recessive gene. No matter what, the mom is going to donate A allele. So no matter what, it's about 100% chance that their children will be A blood. Last bit is called polygenic inheritance. And polygenic inheritance is when more than one gene controls a trait. For example, skin color and eye color, and we produce a range of phenotypes. And so we'll actually look at a lab here where you have, this is an example of 3 different alleles that would control eye color, skin color, hair color. Height is another example. Just multiple or more than one gene would control this particular trait. So we actually have a lab that goes into that. So backing up a bit. So this really concludes the notes. It's been about 20 minutes, and just make sure that you come to class with the notes complete so we can do some more practice problems. We are going to do some practice problems with dihybrids, with the blood types, with the incomplete dominance, the sex-linked traits. We will be completing quite a few of those Punnett squares and doing a couple of labs in between. So make sure you have these notes complete before you come to class.